Okay, and thank you, Norm, for having us today. And I'll be making some forward-looking statements. We are a public company. Move right into our pipeline here. So Fibrocell, if you're not familiar, is focused on rare skin diseases, primarily in pediatric subjects. And we do this using autologous fibroblasts as a platform to carry genes of interest to the skin locally and treat these diseases. And we have a couple of items in the pipeline I'll talk about today, but I'm going to be focusing today, with the time we have, on our main product, which is FCX007, which is furthest along in the clinic. A little bit about our pipeline here. FCX007 is for the treatment of recessive dystrophic epidermolysis bullosa. This is a rare skin disorder that's caused by a mutation in the COL7A1 gene. So collagen 7 is the culprit here. It creates an anchoring fibril that binds our layers of skin together, and then when that is malformed or if it's null, these patients have blisters all over their body, and I'll get a little bit more about the disease shortly. I will not be talking about this in detail today, but we also have a second program in localized scleroderma called FCX013. This is sort of the opposite idea. We have a gene therapy here where it's a autologous fibroblast that's modified to hyperproduce MMP1 to break up sclerotic disorder. So we really see this as a platform on its own, with our first indication in localized scleroderma, which is currently in the phase 1 portion of the phase 1/2 trial. So moving into our platform. So, this is an autologous approach, so we are collecting small skin samples from patients. We're growing them up in our GMP facility in Exton, Pennsylvania, which is outside of Philadelphia, which I'll talk about shortly. And we're using lentivirus in order to create our gene modification. Some of the reasons why: it's mainly about packaging capacity. A lot of these ECM proteins, the gene constructs, for instance, for COL71 is 9 kb. So really, the payload is really efficient for some of these larger genes for proteins. And also, the integrative nature of lentivirus is beneficial in a disease like EB because of the chronic nature, so we do want to see prolonged protein expression. And then what we do is, once we complete the transduction, these cells are grown up, and we create a personalized bank for each patient. And so that gives the ability for these patients, you think of chronic disorder in skin, we're treating a region locally and we hope to have this long-term effect there. There'll be an existing bank for these patients to go back to and access over the course of their lifetime. And then we do all this in our GMP facility, like I mentioned, outside of Philadelphia. We have a cell therapy facility that we've had in the company for quite some time now. It was actually part of an approval for a previous BLA that we had back in 2011 in the field of aesthetics, but now retrofitted now for our ex vivo cells that are gene modified. This facility is 13,000 square feet. We have clean room space and quality labs, and we have distribution set up for autologous lots and have a lot of experience with cold chain and able to validate that coming into our new programs. And we also have both programs, both 007 and 013, coming through this facility. So it is in our INDs as a multi-product facility as well. And of course, we have all the quality systems, logistics and tracking, and raw material and inventory control upset. So this will also be the location of launch of our main program, and we already do have capacity to service the US or the entire RDEB market. So getting into our program, so FCX007, we do have a number of designations, as many of our rare diseases that we work on do. We have orphan drug. We also have rare pediatric disease designation for this particular indication, so obviously it's voucher approvable upon approval, it's voucher eligible. And we do have fast track. The most recent news we've had just a few months ago now is we did receive RMAT designation for this. So one of the, Dr. Marks was talking today about the RMAT program and how there's, a lot of new access to that with 44 approved, and we were one of those this year. We've also had some access to grant money as well. So between the FDA's orphan drug department, and also one of our advocacy groups who do grants, we've been able to fund this program in the past through some other avenues as well. So moving back into EB, and if you're not familiar with disease, luckily over the last few years, EB has been receiving more and more media attention and a lot of advocacy support, which is wonderful because obviously this is a very horrible disease. So what you see in these pictures here are these patients who express these just terrible lesions all over their bodies. They have wounds, they either can be chronic, non-healing, they can reoccur over time, and as you can imagine, not only the pain and the kind of lifetime of managing this disease, their life expectancy, it's a very high mortality around the age of 30 because squamous cell carcinoma becomes an issue, obviously, because you can imagine sepsis is an issue. And there is no approved treatments for these worldwide. And really the state of art right now is palliative care. It's really bandaging. There's a lot of sepsis baths where they have to use bleach in order to keep sepsis down on the skin, and really feeding tubes and surgeries for esophageal dilatations and finger separation surgeries because they can bind is really state of the art right now. So back in May of last year, 2018, our PI, Dr. Peter Marankovich from Stanford University, presented some of our initial phase 1/2 data. We ran an open label intra-patient controlled study. So if you imagine we would have a wound on a patient, it would be locally treated, and then it would be matched to an untreated wound on the same patient in order to collect some comparability data. And what we did with the FDA's agreement is we started in adults. Then after we collected some safety data and some benefit data, we then petitioned the FDA to move into pediatrics, so that's where our phase 1/2 split occurred. But what we presented were in 5 adult patients ranging from ages from 20 to 37, 8 wounds treated. So as you can see, a few of the patients received more than 1 wound treated. The goal is to treat as many wounds as possible, and of various sizes. And we've actually since then have treated even larger wounds than that, and working on some dose ranging as well. And so our patients, although these are intradermal injections, so if you imagine the wound, what we're doing is implanting these cells around the periphery of the wound. The hypothesis there is it's facilitating wound healing while laying down new collagen 7 in order to promote reepithelialization of the wound. We did these actually outpatient. We don't need surgical intervention for this under general anesthesia. It was very well tolerated, the process itself. And we've been looking at safety, obviously, primarily in this study out to a year. And out 52 weeks, we didn't see any serious adverse events or product-related AEs. We didn't see any replication-competent lentivirus results. Obviously, we had to do RCL not only on our product and the vector, but the patients themselves. And most importantly, there were no autoantibody responses. So as you can imagine, you're reintroducing this protein into patients. There's always a potential of eliciting an immune response. We've done localized testing and systemic serum testing, and luckily we have not seen any issues. So since then, we have dosed a pediatric patient. We actually enrolled our full phase 2. 7 patients were enrolled. We dosed 2. I'm going to go through a little bit of the data, but in that, we've actually have had some FDA interactions that have moved our program along that I'll share. And what's become sort of in the EB space an important endpoint is obviously, as you can imagine, is wound closure. And one of the endpoints we looked at from an efficacy standpoint were, what does the degree of wound closure look like for these patients? And as you can see in the top graph, after only a single injection of FCX007, out of the wounds treated, you can see it's greater than 50% healed. These are measured by the PI, 75%, and then importantly, complete wound healing. And as you can see, at complete wound healing, obviously we saw a significant rate of closure versus untreated wounds. The reason why we're highlighting this in our data is we have just recently completed an end of phase 2 meeting with FDA. We did a Type B live in Silver Springs, and part of our discussion was around what's the primary endpoint for a pivotal trial. And in the end, this complete wound closure FDA believed was an acceptable primary endpoint that they can be comfortable with because of things like the previous wound guidances that exist and also the ulcer guidance. And then, of course, the new EB guidance that came out this year and was finalized. So this will act as our primary endpoint. I'll talk a little bit about the design of our pivotal trial based on these data. And then we also looked at some wound closure out. At that point, we only had a few data points out to a year. Again, only on a single injection session, but of course we see a positive trend moving on, and the goal is to see the durability increase as we move along. Now, of course, we wanted to look at some protein expression. Now, in EB patients, it's difficult to collect samples for these sort of testing because you can imagine the skin is very fragile. So we were limited to a few samples, a single biopsy per visit or something of this nature. But what we were trying to do here is look at skin cross-section and determine, are we seeing collagen 7 being deposited in what's called the basement membrane zone, which is the space in between the skin. And if these collagen 7 fibers form up into an anchoring fibril, if you think of it like Velcro, there's the hoop, and then there's the lattice below. The lattice is your collagen 1 and 3 in your dermal layer, and the hoop, think of that as the as anchoring fibril. If you imagine if that was broken or not there, those Velcro layers won't stick. It's the very same mechanism in skin. Here, as we see in the middle panel, that's an antibody to collagen 7. And in 2 example patients, we saw out at 25, 52 weeks, and 4 weeks, 32 weeks in another patient. Interestingly, it appeared that it was increasing over time. So, we actually collected some PCR samples as well. We can see the insert from the vector, out at least 6 months, and obviously we see some protein data out a year. So we believe that the autologous approach, especially in kids who have, highly sensitized and, sort of always in an inflamed immune response state, is the right way to go for this sort of therapy. So just a little on our phase 3. So we did hold an end of phase 2 meeting, as I mentioned, back in a few months ago now, in April with the agency. We finalized this design with them. It's going to be called Defy RDEB, stands for dermal fibroblasts, RDEB. Again, it's a very similar design to our phase 1/2. It's open label. We'll be doing intrapatient design. Very importantly, we will be pre-monitoring these wounds that we've done previously for 3 months minimally. We want to ensure these are chronic, non-healing wounds. The feedback is if they're self-healing wounds that can cycle, that can create a bias in our analysis. So we are pre-monitoring these wounds to ensure they don't close. And we're going to be treating up to 3 pairs of wounds per patient. And we're going to be giving 2 doses of FCX007. We've done our escalation. We're going to do one at baseline and another one at 4 weeks to act as sort of like a booster. If we can close the skin and create a better environment, we hope to create more and more collagen 7 and have that move out. So we've agreed on a 15 to 20 patient trial. We'll be doing this at Stanford University, Colorado Children's, and we're looking at some other, so you can think about geographically, some other universities as well, only because travel is very difficult for these patients. Primarily looking at the comparison proportion of the 007 treated wounds to untreated, and we have agreement with FDA that these will be untreated match wounds due to the impacting condition on skin at 12 weeks post initial injection. Secondarily, we'll look at 50% wound healing. We're going to look at a patient report and outcome measure, and we're also going to look at durability out to 24 weeks. And then exploratory endpoint will be a subset. We'll look at some additional collagen 7 expression, although it was agreed that obviously the clinically relevant endpoint was of interest. Okay. So we have filed this protocol to the FDA. We had some remaining CMC items. They were all filed in July. So we were technically active on this. We're active on this pivotal trial, and right now we're going through the IRBs, just completing that cycle because you have that after FDA filing, and we hope to have a patient in shortly and kick off the enrollment and through 2020 plan to do dosing and a lot of our, obviously our enrollment, manufacturing and dosing, and then have been targeting in 21 for a BLA on this program, which we also discussed with FDA what the structure of that should look like as well, including sections for vector manufacturing and also our cell process and obviously our clinical outcomes. So we're very excited. This program has been moving quickly this year. We also, obviously I mentioned RMAT and we have some of the other scale up that we've been doing to get to this point. So I thank my team for that. They've done an amazing job with a small group, and we're really looking forward to a readout on this in the next year or so. Okay. Thank you very much.